Hi everyone, I'm here today in the SciTech Student Learning Lab, and I'm about to do an investigation that's all about photosynthesis. Before we dive in, what is photosynthesis? Photosynthesis is a process in which plants use carbon dioxide and water to create carbohydrates, or sugars. During this process, oxygen is also produced. Today, I'm going to try to observe this process under two different conditions, light and dark. I'll start by making sure I have all the materials I need. I'll be using water, Elodea, which is an aquatic plant, 4 test tubes with caps, 2 250 milliliter beakers, and a grow light. I'll also need baking soda and a cardboard box. Once I have all my materials, I can go ahead and add 1 tablespoon of baking soda to the 800 milliliters of water I already measured out. The water is regular tap water, and the baking soda is the exact same stuff you might use at home to make cookies. The scientific name for baking soda is sodium bicarbonate, and in our investigation today, it's going to serve as a source of carbon dioxide for our plants. Remember, carbon dioxide and water are 2 of the essential components needed for photosynthesis. I'll pour 200 milliliters of this water into each of my 2 beakers. I'm doing everything twice because I want to test 2 different conditions, light and dark. So one of these beakers is going to eventually end up in the light, while the other one will be in the dark. Now I'm going to fill up 2 of my test tubes with water and invert them, or turn them upside down in my 2 beakers. I'm making sure to fill the test tube completely to the top. Even spilling a little, that's fine. Then I'm tightly screwing on the cap and inverting the tube. I'll remove the cap when it's underwater. Why go through this whole process instead of just flipping the test tube upside down? Well, I really want to make sure that there's no air in the test tube to start out. It should be completely full of water. These test tubes are our controls. They show us what happens when there's no plant at all. That's why they only have water in them. I'll speed up the second one since you already know the drill. Next, I'll add our plants to my other 2 test tubes, then fill them with water and repeat the process of getting them inverted in the beakers. Again, it's really important for me to make sure the test tube is 100% full of water. Well, water plus the plant. There should be no air at the top. So if I spill, that's okay. I'm just making sure that there's no air left in the top of the tube. I'm doing the same thing a second time so that each beaker will end up with 1 control test tube with just water in it, and 1 test tube that has water and a plant. So far, we're keeping everything the same between the 2 beakers, but that's about to change. Now it's time to bring in the light. I'm going to choose one of my 2 beakers and set up a grow light so it's shining directly on the 2 test tubes in that beaker. A grow light is an artificial light that mimics the light of the sun. I need to put my other beaker in a dark place. To make this happen, I'm going to use a recycled cardboard box. I've added duct tape to cover any holes or cracks where light could get through, so my beaker will be completely in the dark. Now we just wait and observe. We can't really look at the beaker under the cardboard box without exposing it to light. So for now, we'll take a closer look at the beaker in the light. If you watch closely, you can see small bubbles of gas coming from the plant. They come off the leaves and then float up to the top of the test tube. Let's slow that down and watch one in slow motion. What's in that bubble? Well, it's oxygen. When plants photosynthesize, they produce oxygen along with the carbohydrates they need to grow. So we're watching photosynthesis happen here. I'm not seeing any of those bubbles in the tube with just water, and that makes sense. If the plant is what's actually doing photosynthesis, we wouldn't expect to see that happen in a tube with no plant. I'm going to leave these plants here to do their thing for an hour. Then we'll check on both the light and the dark plant and see if there are any changes. So here we're looking at the plant in the light, and it looks like quite a bit of gas has accumulated at the top. Remember, that's oxygen produced by this one tiny plant over the last hour. The test tube with just water has a little tiny bubble. It could be that some gas has been produced, or it could be that I let a little bit of air in when I was inverting the tube. Now let's look at the beaker that was in the dark. To start out, I'm not really seeing an accumulation of gas like what we saw in the other one, even in the test tube with the plant in it. Both test tubes here have stayed the same. So what does that tell us about photosynthesis? What conclusions can you draw from this investigation?